Welcome, my dear students, to another week of Biology with Mr. Miguel. This week we will continue speaking about human nutrition. Our main focus today will be enzymes. These are the objectives for this week, so let us delve straight into enzymes. When we speak about enzymes, this would be how enzymes would normally look like. If you notice, it looks like coils of wire and cinta, ribbon, bonded together. After the computer has processed the image of, or the three-dimensional image of an enzyme, this is how it would look like. For simplification purposes, in this biology class, this is how enzymes will be depicted. Only conceptual. So only conceptual diagrams of enzymes will be depicted within this presentation and in this class. So what are enzymes? Enzymes are proteins, first of all, that serve as biological catalysts. Note that the word catalyst is bolded in red. So what does the word catalyst mean? The word catalyst refers to any substance that is able to accelerate a reaction. That is exactly what a catalyst is. So a catalyst is anything that will help you to go faster and make your life easier. An enzyme is a biological catalyst, meaning that it's a chemical that will help accelerate the chemical reactions that occur within the body. But enzymes work in two different ways. Sometimes enzymes are used to break down substances, like you have a very large substance, an enzyme can be used to break down that large substances into smaller substances. Some enzymes can also be used in building large substances. Maybe you have small little pieces and some enzymes are programmed to build larger substances from simpler substances. So it can go either way. Some enzymes break down, some enzymes construct. That is my main goal here. And there are thousands of enzymes within the body. Not only the human body, we also have fungi, we have plants, we have bacteria that utilize enzymes to carry out the reactions that are occurring within their bodies. Because our topic is human nutrition, we want to focus on the enzymes that are found within the digestive system of a human. In your mouth, you can find a very common enzyme which we know as salivary amylase. An amylase is used to break down complex sugars such as starch. So amylase breaks down starch, which is a carbohydrate. If we move a little bit down, we reach the stomach. In the stomach, we can find an enzyme that is charged and responsible with the breakdown of protein. The name of that enzyme is pepsin. We can also find in very young mammals another enzyme. This enzyme is used to break down the proteins found in the breast milk. And the name of this enzyme is renin. Now here is the fun part. Milk is a mixture of sugars, proteins, and fats, and of course water. So whenever you drink milk, or when you were a baby and you used to breastfeed, the milk that you were consuming contained carbohydrates, proteins, and fats, and of course water. But the enzyme, when you were a baby, that used to break down the protein in milk is called renin. The one that would break down the lactose, which is a sugar, a carbohydrate in milk, was lactase, or is lactase to this point. So you have different enzymes in charge of breaking different substances down. Let us move into the small intestine. In the small intestine, you're able to observe that there is various types of enzymes being dumped into the small intestine. For example, we have chymotrypsin, which breaks down proteins. We also have trypsin, which also breaks down proteins. We have amylase, which breaks down carbohydrates. We have lipases, which break down lipids or fats. So in the digestive system, normally we will find digestive enzymes which are responsible for the breakdown of large food substances into smaller food substances that can be readily absorbed into the body. So let me talk a little bit more about the process of breaking down and the process of building. When we look at all the chemical reactions happening in your body, there's millions of chemical reactions happening every single second. Some chemical reactions are breaking down substances, other chemical reactions are building substances. The specific group of chemical reactions that break down substances is known as catabolism, like the word kata in catastrophe. Catastrophe means like it's a tragedy, it's a breakdown. This is more or less how I would relate catabolism with catastrophe. They both start with the prefix kata. Catabolism is the reactions, the chemical reactions that are breaking down the substances, the large substances into simpler substances. Let me give you an example. In this case, we have an enzyme. So this, where I'm pointing at, this gray blob, that is our enzyme in this case. And the green is the substrate. Please take a brief pause to observe the different structures that are found in the enzyme. You notice that on the enzyme there is a landing site that we know as the active site. This is where all the chemical reactions that this enzyme will perform will occur. When the substrate approaches the active site of the enzyme, it binds with the enzyme. So this is number one. Look at number two here. In the second little diagram, you're able to see that the substrate, which is the chemical or the substance that will be broken down by the enzyme, binds with the enzyme. Once it binds with the enzyme, it is broken down by the enzyme. So the enzyme serves like a little knife that will cut this large substance into simpler substances. Once it has cut this large substance into smaller substances, it will release the products. In this case, we see two products being released. So you start with one substrate. This substrate is the large substance which will be broken down. And once it's broken down, the products are released. Notice that the word catabolism refers to the group of reactions that are occurring inside of the human body that are breaking down substances into simpler substances. Now let's look at the other side of the story. Now we will move into anabolism. And the prefix ana means to join or to bring together. And anabolism is the group of chemical reactions that join simple substances into a large substance. It means like imagine Legos being used to build houses, used to build cars, and whatever else you can build with Legos. Small little pieces or small little substances will be brought together by the enzyme, joined by the enzyme to build larger substances. Let us look at the example here. The gray part represents our enzyme. In number one, we have two substrates. Find two substrates. These two substrates will approach the enzyme and land on the active site. Once they land on the active site, they will bind with the enzyme. The enzyme will join them, normally through a process known as condensation. And once they are joined, the enzyme will release the new product, and the new product is a larger substance than the original substrate. This is known as anabolism. Anabolism is the group of reactions occurring within the body that is used to build larger substances. Enzymes obey a theory that is known as the lock and key theory. This theory basically tells you that one key corresponds to one lock. In this case, the lock represents the enzyme and the key represents the substrate. What does this mean? It means the following. If you have a substrate that the enzyme is not designed to hold or work on, nothing will happen. If you give amylase a protein, a piece of meat, nothing will happen. Why? Because the substrate, which is the meat, cannot bind or lock with the enzyme, and if this cannot happen, then there is no chemical reaction. This in itself is known as the lock and key theory or hypothesis of enzymes. Apart from being highly specific, enzymes are extremely sensitive. That means you cannot scold an enzyme, tell this enzyme, hey, you did wrong, because the enzyme will feel sad. I'm just kidding. When I say that enzymes are extremely sensitive, I'm talking about temperature. Increase the temperature a little bit or decrease the temperature from the optimum temperature of the enzyme, and the enzyme will not want to work. Change the pH. pH means the acidity. Change the acidity of the enzyme and the enzyme will not want to work. Change the quantity of substrate that is in contact with the enzyme will not want to work. So enzymes are very sensitive. Anything can get them really sad, and when they're sad, they don't work. So let us look at the first one. Enzymes are sensitive to temperature. At low temperatures, the enzymes cannot work fast enough. This means that they work very slow. Why? Then look at the screen. This is one enzyme here. When you have very low temperatures, what happens to the movement of particles? Particles cannot move freely or cannot move energetically when it's too cold. Okay, so particles, imagine that I'm a particle and it gets too cold. Instead of me moving a lot, I will remain in place and freeze to death. That's possible. So in a way, when the temperatures are too low, the enzyme is unable to work because the substrate is unable to move fast enough to reach the enzyme. So these are the substrates. In low temperature, they will move very sluggishly. Okay? What about high temperatures? At high temperatures, enzymes denature. Enzymes are proteins, and proteins tend to denature at higher temperatures, and that is why enzymes are unable to work at temperatures that exceed their optimum temperature. The temperature needs to be just right. For humans, all the enzymes work at a temperature of 98.6 degrees Fahrenheit, and they work well within. Now, when you have a fever, then the enzymes kind of slow down because the temperature is too high, and this causes the enzyme to denature. And if you're too cold, the enzymes also work slowly because the substrate cannot move fast enough for the enzyme to work on it. I want to focus a little bit on enzyme denaturation. In this slide, you're able to see two diagrams. The first shows an enzyme that is in contact with a substrate. Notice that the active site of the enzyme matches and locks with the substrate at normal temperature. Now, if you increase the temperature, the shape of the enzyme, since it's a protein, will change dramatically. Once it changes dramatically because of the temperature, the substrate will be unable to bind with the enzyme, and if it's unable to bind with the enzyme, there is no net chemical reaction occurring. And that is how high temperatures cause denaturation and affect enzyme activity. This graph shows exactly what I was talking about. At the middle, you find a line. This blue line is known as the optimum temperature. So you go way down and you notice that there is a temperature at which the enzyme works best. When you increase the temperature, that means that you move towards the right. What happens to the enzyme activity? It starts to decrease. So on the y-axis, you will find enzyme activity. You notice that at very high temperatures, activity becomes low. Let me just show you what I mean. You notice that at high temperatures, the activity itself is very low. If the temperature is too low, that means that it's cold, the enzyme activity is also cold, it's also low. Notice. And that is because the substrate is moving too slow for the enzyme to work on it. Do you understand what I'm saying? At optimum temperature, the optimum temperature is the temperature at which the enzyme will work the best. It is where the enzyme says, yay, let's get this party started. And that is what is happening at the optimum temperature. And this is how temperature affects enzyme activity. Let us watch this short video for you to clearly understand what I'm saying.
Environmental effects on enzyme function. Active sites are very sensitive. They sense even the slightest change in the environment and respond accordingly. Some of the factors that affect the active site and consequently enzyme function include temperature. The suitable temperature for enzymes to function properly is 37 degrees Celsius. Increasing or decreasing the temperature above 37 degrees Celsius affect chemical bonds in the active site, making them less suited to bind substrates. Higher temperatures denature enzymes.
Enzymes are also sensitive to pH. The little acronym pH stands for potential hydrogen. And potential hydrogen means this. If you know anything about acids, is that acids are basically hydrogen ions within a solution. That is what an acid is. An enzyme is unable to work outside of its normal pH. This means that every enzyme is designed and programmed to work at a specific pH. There is something known as a pH scale, and the pH scale shows the acidity from 1 to 14, 7 being neutral, it's not acid nor alkaline. Some enzymes work marvelously at a pH of 1, and some enzymes work wonderfully at a pH of 14. Depending on the enzyme, they will have different pH at which they work best. And a pH that is outside of their normal operational environment would result in a denaturation of the enzyme. Once more, let me show you what this means. In this image, you're able to see, in the first image, you're able to see a normal enzyme working at their normal pH. In the second image, you're able to see that the enzyme has been denatured by maybe acid or maybe a base or alkaline, and the shape of the enzyme changes. If the shape of the enzyme changes, it is unable to bind with the substrate. If it is unable to bind with the substrate, bam, it doesn't work. So enzymes cannot tolerate big differences in pH. Let me show you the graph and let me illustrate a little bit of what exactly I mean. In this graph, I am showing and illustrating 3 different enzymes working at different pH. Each enzyme is designed to work at a specific pH. From example number 1, which is pepsin, you're able to see that this enzyme works best. So the activity of the enzyme works best at a pH of 2. That is where the enzyme will work best. Now, if you notice, if the pH gets to 4, that means it gets a little bit too basic for the enzyme or alkaline, the enzyme activity decreases substantially, that it's nearly non-existent. This means that this enzyme will not work at a pH of 4. In comparison, you have another type of enzyme which you can find in your mouth, which is known as salivary amylase, and this enzyme works best at a pH of 7. How do I know? Because the enzyme activity is the highest at a pH of 7. If it gets to acid, what happens? Let's say it gets to pH of 5. What happens to this enzyme at a pH of 5? The activity of the enzyme decreases dramatically. What if it increases the alkalinity? That means it gets to a pH of 9. So at a pH of 9, the rate of reaction for this enzyme is nearly non-existent. What I'm trying to bring across is that enzymes work at very specific pH. If you change the pH of the enzyme, it will decrease its activity.
pH. Amino acids present in the active site are acidic or basic. Fluctuation in pH can affect these amino acids, making it hard for substrates to bind. Extreme pH values can denature enzymes.
On this final slide, you're able to see that enzymes are sensitive to substrate concentration. When we talk about substrate, it is talking about the substance with which the enzyme works with. It means the substance where the enzyme will work. That's what it is. If there is more substrate, the enzyme will work very quickly. If there is little substrate, the enzyme will work very slowly. In these two diagrams that I'm trying to portray here, the activity in this enzyme, so this gray box represents an enzyme, and these little circles represent the substrate. In this scenario, we will have a very slow reaction. In contrast, in this scenario, we will find that the rate of the reaction is much faster. Why? Because there is more substrate. This one is a little bit of a no-brainer. It doesn't require you to think that deeply about it. The more substrate there is, the faster the enzyme will work. One final thing, enzymes are not used up in any chemical reaction. This means that an enzyme can be reused over and over and over and over again.
Substrate concentration. Increasing substrate concentration increases the rate of reaction. This is because more substrate molecules will be colliding with enzyme molecules, so more product will be formed. But again, this effect is valid up to a certain concentration.
Thank you very much for your keen attention. Have a blessed rest of the week.